So today I'll be talking about the electron transport chain and what our processes kind of go into that. So first, the electron transport chain is located within the inner membrane of mitochondria. So basically this chain is a chain of redox carriers. So first, NADH and FADH2, which are electron carriers and products from the citric acid cycle, are used in this process also. So first, NADH is used and donates its electrons to the first redox carrier, which is NADHQ reductase. And through this process, hydrogen ions are pumped from the matrix of the mitochondria to the intermembrane space. And so the two electrons, or the electrons that were donated from the NADH, are then moved through the electron transport chain to the coenzyme Q, which is the next redox carrier. And when that NADH also was used, it donates that electron, it turns into NAD+. So those electrons in the electron transport chain are now in the coenzyme Q. So here, with this process, FADH2 is then the energy is used and produces FAD. So then those electrons from the coenzyme 2 are moved down the electron transport chain into the third redox carrier, which is cytochrome reductase. Cytochrome reductase, and here another hydrogen ion is pumped from the matrix of the mitochondria to the intermembrane space. So those electrons continue to be pumped to what is called the cytochrome C, which is the next redox carrier, and then are continued to move down the electron transport chain to the cytochrome oxidase. So by the time these, and also here, a hydrogen ion is pumped from the matrix to the intermembrane space. So by this time, the electrons that have been transferred through the electron transport chain have lost most of their energy. So these electrons have to be put somewhere. And the last phase of the chain is these electrons are placed with oxygen. Oxygen is the electron acceptor. And from there, as I was said before, hydrogen ions have been pumped from the matrix of the mitochondria to the intermembrane space. And with that has caused a high concentration of hydrogen ions in the intermembrane space. And with that, to create ATP in this process, ATP synthase is used, where hydrogen ions flow through this concentration gradient, and in the process, three catalytic sites are used to in the ATP synthase are used to join ADP and inorganic phosphate, which ultimately create ATP in this process. Thank you.